few slides to sort of try to touch on some of the aspects of fundamental research that might be of interest to you. But I'm more than happy for anybody to stop me at any point if you're not understanding something or if you have a different question that I'm not covering, so as interactive as possible, because I feel that my main reason to be here is to help you understand the neuroscience underlying SYNGAP1. So, as Andy mentioned, I'm the Director of Fundamental Research. I direct the Patrick Wild Center with Andy, who's the Director of Clinical Research. We also have recently, at the University of Edinburgh, started the Simons Initiative for the Developing Brain, which is all funded by the Simons Foundation Autism Research Initiative, which probably many of you will know about. It's based out of New York. And it's the biggest philanthropic research institute in the world for things like SYNGAP1. I also, science is very international, so I also am the Associate Director for the Center for Brain Development and Repair, which we have in India, which is a sister center to the Patrick Wild Center and is led by Shona Chatterjee. And really I have to put up all three of these affiliations because the work I'm going to show you today, the small bit of work I'm going to show you today actually comes from all three of these centers. So what I thought I'd do is give you the overall concept of how we approach starting to understand these monogenic neurodevelopmental disorders like SYNGAP1. And actually SYNGAP1 is one of our largest programs of work here at the University of Edinburgh. But we study a range of these monogenic forms, and I'll try to get across why it's important to not just study one. I think you got that probably from Nick's talk, etc., about how much we can learn from other disorders, even if it's not the same gene. I'll try to highlight bits of that. This is the team here at the University of Edinburgh, that sort of the fundamental research and clinical team. Let's see. A much younger Andy down here. Noboru Komiyama is at the back of the room, and I'll be talking about the mice that he generated and how we use them. But really, many of you might know David Fitzpatrick, who's a key member of the DDD team in the DDD study. And we have quite a good team. I'm also going to talk about some of, in the top middle, that's Adrian Bird. I'll be showing some of his groundbreaking work. And that's the director of the center out in Bangalore, Shona Chatterjee. But everybody on this slide is working on different forms of monogenic disorders and trying to understand brain function when a gene goes wrong. So this is our center out in Bangalore, and I often show slides of Edinburgh, and it's nice and sunny out, which is basically not very often. It's been a lot. But in Bangalore, so it is actually always sunny, and you can see we have a very big team down in Bangalore also working on these disorders. So what is it that we're studying? So we study, as I sort of alluded to earlier, monogenic forms of severe neurodevelopmental disorders. And I'm sure, as Mick mentioned earlier, unfortunately, I have to say I'm meant to apologize for not being here at the beginning of this conference. The CDKL5 family conference is also going on in Edinburgh today. So I was there this morning. Some quite similarities between CDKL5 and SYNGAP in terms of the severe seizures and in terms of the intellectual disability. So I was there this morning and rushed over here to come be with you. So we studied where all of these interlap. So where the kids will have autism, they'll also have intellectual disability, and they'll also often have childhood epilepsy. So SYNGAP would fall right in here. And what the DDD showed was that if somebody presents with one of these disorders in the clinic, about 60% of them will be discovered to have a monogenic disorder. So there'll be a single gene cause for that. And if you look at that and you overlap it with the big genetic studies that the Safari, the Simons Foundation, has done for autism, there's a huge overlap between the genes that come out of the DDD study and the genes that have come out of all of the Simons study. So really we're all studying the same thing. So it doesn't matter whether you call it autism or intellectual disability or childhood epilepsy. In many cases it's the same thing caused by the same genes. Now, when you look at all of these genes, and there's probably, if you go by the Safari list, there's over 100 genes now, that if something goes wrong in that gene, you will end up with a severe neurodevelopmental disorders. And quite often people go, oh my God, there's 100 genes. If you look at the risk genes, goes up to 1,000. And you think, oh my God, does that mean we have to come up with 1,000 different therapies? And the real answer is we don't know, but there's a lot of reason to hope that that's not true, and in fact that we can break these disorders down into smaller groups, and that might serve common treatments. And that's essentially what this slide is showing. This is from one of the Safari investigators from the big genetic studies, and if they cluster the genes that come out of these big screens, you can see that they sort of congregate around specific functions. So either synaptic function, which are the points at which neurons communicate with one another, or chromatin remodeling, that's where Rett syndrome and other disorders like that, CHD8, will fall into, or the Wnt signaling pathways, which is a very particular type of biochemical pathway in the brain that regulates brain development. And so if you look at these, you start to get a bit of hope that actually, since these are all sort of congregating on common functions, maybe the treatments will be similar. And you can see SYNGAP1 right in there. And you can see that the size of the circle in this diagram represents the number of cases at the time from this study. So you can see that CHD8 is probably the most prevalent of the monogenic forms, but SYNGAP is right up there. So the questions we try to address in the lab is, does this what we call genetic convergence, indicating the common potential functions, does this actually represent convergence in how the brain goes wrong during development? And how do we go about doing this? In many cases, people call this the virtuous circle, but I refer to it as the potential of molecular medicine. So how does understanding the genes and the molecules that go wrong during brain development, how can we turn that into potential clinical therapies? So for human neurodevelopmental disorders, as Mick I'm sure has gone through, we start with gene discovery, then we start to make preclinical models. And preclinical models can vary from flies to fish to mice, which is probably the most common, to rats, to now non-human primates, big animals like pigs, and then through to humans. And with the big change in our ability to edit genomes, that we used to be stuck with the mouse, and I'll come back to that, but now we can make, with these new gene editing techniques like CRISPR that I'm sure many of you have heard of, we can start making whatever animals we want. And at the Patrick Wild Center, at the Simons Initiative for the Developing Brain, we've moved very heavily towards rats, and I'll show you some of that data coming up. But the key thing about this slide is what we do once we have our preclinical models is we really try to understand the basic neurobiology and the disease pathophysiology. We then can come up with theories for how we might intervene. And then we go back into our preclinical models and test those. And I wish it was we just had to do this once, but typically you learn a lot from that and you go round and round and round and round. And this can take a very long time, right, before we actually know about a gene and then before we can go into a clinical trial. So I've done a lot of work over my career in Fragile X syndrome. The gene was discovered in the first mutant mouse was discovered in 1993, or was made in 1993, and the first clinical trials didn't happen for 25 years with the young trials. So this can take a long time, but we're getting better at it. We're learning. We learned a lot from those trials in Fragile X, and we won't make those same mistakes again. Or Andy and his colleagues who run clinical trials are unlikely to make those mistakes again. But we learn a lot as we go along, and even when it comes down to how to do the best clinical trial, a lot of these things are already being done. And many people have asked me about gene therapy. The gene therapies are already being done and are in the process for Rett syndrome things, and we're going to learn a lot from those. So by the time we might be applying any of these types of clinical trials to SYNGAP, by going through it with the other monogenic disorders, hopefully we will have learned a lot by then, and they'll be much more efficient. So how do we go about doing this in the lab? So there's the classic approach in science has always been a bottom-up approach. So if you understand the genes and what they might do in a synapse or what they might do in a cell, then you can start asking questions. You move up to the cells, and you might say, how does that change the physiology of a neuron or a brain cell? Then you try to understand the brain is a big, wide circuit. How does that affect circuit function? And ultimately, how does that lead to behavior? And that is a classic sort of reductionist approach of science, from bottom up. But we've taken a much different approach as well. So we do the bottom-up approach. Many of the people you saw on that first slide will be bottom-up people. But many of the scientists now in the center are using a top-down approach. where we start with trying to understand the animal's behavior, and we work backwards. We then try to understand what goes wrong with the circuit, and hopefully we'll meet in the middle. So by the time we are testing something where theory came from a bottom-up approach, we already have robust behavioral or circuit-based alterations that they can use to test their intervention. But there's another really good reason, because for neurodevelopmental disorders, and I'll come back to this in a minute, we actually don't know whether there's critical period time windows during development when treatment might help, if you go by the theories that come from bottom-up approach. But if there is critical periods, you might miss that window, okay, when treatment is most effective. Top-down approaches bypass that, because they look at what's wrong in the brain circuit and see if we can tweak the circuit a little bit to get a better output. So they're not, I wouldn't call these types of approaches cures, but they might be able to help with particular symptomatologies that might be most distressing to the individuals. When we do that, when we take this approach, we don't just focus on one brain area. Probably many of you have heard of a brain region called the hippocampus, because it gets the most attention across neuroscience. But actually we know the brain is more than just the hippocampus. So we try to study these in a range of brain areas. And what's absolutely key is we need to understand what happens during brain development. So we don't just look in adult animals, at adult animal behavior, but we look across development.
Now, just a question.
Yeah.
In analysing the circuit part, if your child's on medication, you can alter the circuit, you have an altered picture, or how does medicine affect it?
So medicines will attack the synapse or some biochemical pathway, but that will have knock-on effects towards the circuit. But what we can develop, and one of the drugs that's been most heavily trialed in Fragile X syndrome and now moving into the autism, is baclofen. Now that attacks a very specific receptor at the synapse, but actually has this huge effect on brain circuits, right? Because it changes the ratio of inhibition to excitation in the brain. So almost certainly, although it came from a bottom-up approach, it's almost certainly having its effect on the circuit. That will just tweak the circuit a little bit, and then the brain adapts around that drug, and the net result is beneficial effects. Does that make sense?
Yeah, well, I don't claim to understand it all, but I think maybe I've talked about it. So if your child's on anti-epileptic drugs,
yes,
does that impact the analysis of that? Or have you answered that question?
Yes.
Yeah.
No. The bottom line is it will depend specifically on what drugs they're on and whether those